Global Community Bio Summit. Thank you very much once again. We have Dr. Karmella, who is already with us to present to us what she's been doing at the University of Emory or Emory University based in the US. So I'll just quickly profile Dr. Karmella. So Dr. Karmella is assistant professor at the Emory University. Karmella Haynes is an assistant professor of biomedical engineering at the Emory University. She earned her Ph.D. in studying epigenetics and chromatin in Drosophila at the Washington University, St. Louis. Subsequently proceeded to do her postdoctoral fellowship at the Davidson College and Harvard Medical School, introduced her to synthetic biology. Her Davidson Harvard Medical School postdoc fellowship project was on bacterial computers, was recognized as Publication of the Year in 2018 by the Journal of Biological Engineering. Today, her research aims to apply the intrinsic properties of chromatin, the DNA protein structure that packages eukaryotic genes, to engineer proteins and nucleic acids that control cell development. After Dr. Haynes joined the faculty at the Emory School of Medicine in 2019, she received an NIH R21 grant in 2019 to develop a new protein engineering and computational tool for cancer epigenetics, and launched the annual NSF-funded Afro Biotech Conference. She's a founder and instructor of the Cold Spring Harbor Summer School on Synthetic Biology, a member of the National Engineering Biology Research Consortium, EBRC, and advisor and judge emeritus for the annual International Genetically Engineered Machines iGEM competition. She has been a featured guest on PBS Nova in 2020 and PRI's A Science Friday in 2016, and her work has been profiled in Forbes magazine in 2020. Let's welcome Karmella to present her work. Thank you.
Okay, thank you for that wonderful introduction. And I'm excited to be here. It's a wonderful opportunity. I'm only a little bit disappointed that I didn't get to travel. I'm not quite ready to travel yet, but thank you for hosting me in spite of that. So I am excited to present my work, Epigenetic Actuation, Engineered Chromatin for Gene Regulation in Triple-Negative Breast Cancer. So before I get into explaining the details of what all of that means, I just want to very quickly introduce you to all of the members in my lab. Without the enthusiasm and creativity and motivation of this group, we wouldn't have been able to get as far as we have been able to get, in spite of some policies and around COVID safety and having to shut down our lab for about 3 months. So we've got a total of 10 members right now. And as I show you some of the results from our work, you'll see some of these faces again. So just wanted to start out by thanking this wonderful group. So my presentation is in 2 parts. For the first part, I'm going to walk you through some of the foundational work that helped us to develop an area that we're calling epigenome actuation. So just a little bit of an introduction to chromatin and epigenetics. And then we're going to segue into how that's relevant to triple-negative breast cancer and what the sort of general impact has, and focused on technology. I'm going to share with you our latest research that is trying to move this concept of engineering chromatin to medical translation. Okay, so, and then just a forewarning, if I freeze a little bit, just give me a couple of seconds so that my internet connection can restabilize. As was mentioned in the intro, my background, right? So I kind of came into synthetic biology as a basic research scientist, sort of focusing on, answering these questions around how are genes regulated, right? Every cell in your body for the most part, have the same set of chromosomes, the same set of genes, but the cellular diversity sort of arises from that single genome. And so what we understand so far is that when we take a look deep inside the cell, inside of the nucleus, and take a look at how the DNA is actually physically packaged, there are slightly different conformations in these shapes that influence whether specific genes along that chromosome are either expressed at high levels or low levels. So back in, I believe it was 1882, a scientist who used dyes to sort of look at these different compartments called these conformations chromatin, or colored material. And so today we've got much more advanced approaches where we now understand sort of the biochemical makeup of chromatin, and we better understand the different parts, the proteins and the nucleic acids that come together to form this chromatin structure. Throughout this talk, you'll see these sort of little cartoons representing these key proteins. Histone proteins are in gray, and then the other regulatory proteins that are not histones will be shown as bubbles. Okay, and then just to give you a little bit more context, I got excited about genetic engineering in general as a high school student, and then decided, okay, well, after I finish college, I'm going to get a PhD because I want to run my own research lab. So this is just a snapshot of some of the work that I got to do as a PhD student. And this is where I really started, I guess, the earliest point where I started combining, recombinant DNA, right? So building DNA constructs and then introducing those into fruit flies as a model organism, whose chromatin structure actually looks a lot like ours in a lot of ways. And then so we were using genetic engineering to plug a what's called a reporter gene, right? A gene that we can actually see, the phenotype very easily. We sort of plugged it into different regions along a chromosome and saw that indeed, these different degrees of packaging influence the expression level of the gene. So we could actually map that out. Okay, so sort of moving forward, by the time I finished my PhD, I started looking for, something a little bit different to do for my research career. And that brought me into synthetic biology. And then so over the years, I had completed a postdoc fellowship at Davidson College and another second postdoc fellowship in Pam Silver's lab at Harvard. And by the time I was ready to establish my own lab, I sort of got excited about this idea of combining synthetic biology with chromatin biology to develop a field that we're calling epigenome actuation. So the epigenome is basically a term for that structure that I showed you in the previous slide, these proteins interacting with DNA to regulate their expression, right? And then you see those changes in expression without any changes in the DNA sequence itself, right? So these are tunable, gene expression levels that happen sort of upon the genome, hence the term epigenome. So today, scientists are very interested in figuring out ways to manipulate the epigenome to change levels of gene expression in order to change the behavior of the cell. It's a really clever approach because it doesn't, work exactly like CRISPR where you're changing the DNA sequence. You're actually just sort of turning up and down gene expression. So this is a commentary that I wrote with my senior research specialist, Natisha, providing an overview of two approaches to epigenome engineering. One is called editing, that changes marks on the histone proteins. And then the approach that we're developing designs fusion proteins that can go in and detect marks on histones and then act upon nearby genes. So I encourage you to take a look at that review, and I'm happy to share by email if you contact me. Okay, so how did we do this? I'm going to walk you through really quickly our approach to building these epigenome actuators that I'm showing you on the cartoon at the bottom right here. So we looked to the natural chromatin machinery and identified an interaction that's very well characterized. And so we used, pretty basic synthetic biology approach where we then went and looked for this protein that mediates the interaction between chromatin proteins and a chemical modification on histone H3 specifically. So the sequence is already known for this protein, and we identified the part that is responsible for this interaction. And then so, like any synthetic biologist, we went and cloned it out, right? So we borrowed it from the natural machinery and then took this bit. And then, so what this protein actually does in the natural system is that when it regulates or when it dials the expression level of a gene, it turns it down, right? So this is a repressor of expression. So we built a synthetic fusion protein that would recognize this histone signal and then turn genes on, right? So we're basically driving the opposite, driving gene expression in the opposite direction with the synthetic system. And then we added an RFP tag so that we could follow the protein around. And so this is an overview of the proof of concept work that we did and had published in 2018. And here we're we've got an artificial reporter gene that's easy for us to measure so that we could basically answer the question, does our synthetic system work as expected? So we actually tried a couple of different versions of this fusion protein, one that just had a single domain for monovalent engagement and one that actually had two tethered together for bivalent engagement. And then what we were happy to see was that not only could we get, some small but significant activation, right, which is what we were expecting on this reporter gene. When we double the domains, it seems to work even better, right? We get even stronger activation of the target that was initially downregulated. So we can switch the regulation from repressed to active with this fusion protein. And so just the last bit of foundational research I want to share with you is that we are, in the context of the whole genome, right, all the entire, 6 billion base pairs of the human genome, there's chromatin all over the place, right? And there's potentially lots of genes being regulated all at once outside of that very simple reporter that I showed you. So the question that we're investigating right now is, which genes become upregulated? And is that one histone modification sort of the only feature of a gene that is responsive to our fusion protein? So this giant heat map, this is a lot, all the genes that we looked at are on the rows. And then we actually we did a time course experiment where we looked at cells that express our fusion protein for 10, 24, and 48 hours so that we could sort of see, get some more, sort of kinetic information on gene regulation. And one of the interesting things that we found was that these genes classify into early responders, sort of delayed responders, and late responders. And we think this has something to do with our protein touching down at certain genes and then activating those, and those genes maybe potentially express products that then go and regulate other genes. So we think we may be able to discover some regulation cascades within the genes that are responding. Okay, and then moderator, just jump in and remind me how much time I have left, just in case, because I know I have more than 15 slides here. So this is basically just an overview of the computational approach we're going to use to make sense out of all of this data, and we're doing that in collaboration with Chris Plaisir at Arizona State University. Okay, so for the second part of my talk, I want to give you an overview of how we're trying to apply this to solve a very hard to treat, or to try to treat a very hard to treat disease known as triple negative breast cancer. So, this slide just gives you a walkthrough of all the technical details, why we call it triple negative. It lacks some key receptors that would make patients good candidates for endocrine therapy, which is an alternative to chemotherapy. We care about this disease a lot. And actually what initially got me interested in it was that I started learning that it disproportionately affects African-American women. And then one of my collaborators at Georgia Tech, Manu Platt, has also been looking into this disease and how it affects women in West Africa, triple negative as well as HER2 positive. So the thing about this disease is that, yes, there's a treatment for it, there's chemotherapy, but chemotherapy is also notorious for causing a serious secondary problem, right? Cardiac problems, right? So your heart cells become compromised, right? They can really suffer some damage from an extended course of chemotherapy. So we really need new approaches to treat this particular type of cancer, as well as other endothelial or solid tumor cancers. Okay, and then I'll just go through this really quickly. So as I mentioned before, the first course of treatment typically for triple negative breast cancer is chemotherapy. At the molecular level, there are some barriers that I believe are, really, a lot of scientists are, starting to realize might help to protect cancer cells from the effects of chemotherapy. So that certainly is a barrier for further advancement and improvement of chemotherapy. And then sort of more advanced idea around treating cancer is, okay, well, why don't we maybe introduce genes, right, that have anti-cancer activity, put those into the cancer cells, express them, and then kill the cancer cells. Well, as anyone who has maybe had some experience moving genes into cells, this can be very difficult in an experimental system, and even near impossible trying to get DNA delivered into a tumor. So there are some barriers there. The approach that we focus on, right, the epigenetic engineering approach, I think is pretty clever, because what we're doing is that we're focusing on the genes that are sitting inside the nucleus. And what we want to do is introduce some sort of manipulation that can target anti-cancer genes that are already in the cancer cell that are just being epigenetically repressed. So if we can turn those back on, then we can express anti-cancer products that are already encoded in the cancer genome, and then stimulate an anti-cancer phenotype to stop metastasis and kill the tumor. Okay, so then some of the data that we've generated so far, we took a look at the public data and then asked, okay, well, all right, this idea, right, sounds nice, that anti-tumor genes are repressed in cancer, and then we can turn them back on. Well, for every type of cancer, you have to ask that question, because gene expression profiles are very diverse from cell to cell. So we went and looked for known tumor suppressor genes. We started with a list of about 580 and narrowed it down, but we still have 99 different tumor suppressor genes that show lower expression in triple negative breast cancer in patients than in some normal comparators. And we also see the same trend appear in cell lines, immortalized cell lines that we grow in the lab and work with. So this is really exciting, because this tells us that we can work with these cell lines as a model system to represent what's happening in patients with breast cancer. So then what is controlling all of this, right? What's causing the downregulation? So a well-known set of repressive proteins is called the Polycomb family of proteins. So PRC is Polycomb repressive complex. This is actually the protein from the cartoon I showed you that we use to build our fusion protein, right? So these repressors are going after tumor suppressor genes in cancer cells and turning them down. And then some data to support that idea. The reason why this happens is that a lot of these proteins, and in particular the two that are highlighted here, are genetically amplified in cancer. There's more copies of the gene, and then therefore more messenger RNA and more proteins. So there's just a lot of this stuff, a lot of this EZH2 protein. And then in the cell lines in particular, a lot of EZH2 and another protein called PCGF, right? So you see this hyper expression of these proteins, and they're kind of going crazy in the cells and turning off tumor suppressors and allowing the cancer cell to be a cancer cell. And this is just a closer look at the structure of one tumor suppressor gene in particular. High levels of this mark here, trimethylation on histone 3, And then the compaction is increased, as is shown by reduced accessibility of the underlying DNA, right? So these black marks would be high if you had like open, active chromatin, but they basically disappear at this tumor suppressor gene. So we have some evidence to show that these polycomb proteins are turning these tumor suppressors off. In order to investigate that a little bit more, we did an experiment where we depleted the cells of either EZH2 or PCGF, right, as is shown by this Western blot. And then what this color scale is showing you is that after you artificially deplete these proteins, a lot of them come up, right? They become reactivated. But I want you to notice that a lot of these boxes are blue, showing that a lot of our key tumor suppressor genes are still shut off, right, when we go in and try a more classical method to perturb this. Then we went in and tried some small molecule inhibitors that bind to either one of these proteins. And we get a lot more genes coming up, but a lot of them, right, the boxes stay blue, right? So this gives us an opportunity to take a closer look and see, or try to use synthetic biology to see if we can improve this, activation of this group of tumor suppressors. And this is just, what I'm showing you is that same CAD M3 from the previous slide, right? So you see good upregulation for the most part. There's another really important tumor suppressor, and this one's very potent, called LHX6. And you see very sort of inconsistent upregulation of that one. So I'm going to show you on the next slide. Forgive me, I'm going to go ahead and skip over the panel on the left for the sake of time. So what we started doing was using one of the immortalized triple negative breast cancer cell lines as a model to further study the behavior of our synthetic activator. And so this just shows that, this is a marker to show us that our transgene that expresses this protein is there. We can use a small compound called doxycycline to control the expression of the RFP marked synthetic regulator. And so that's shown in these panels here. So what I really wanted to get to, because I'm really excited about this result, these are very preliminary data, but what we're seeing so far is that unlike the knockdown experiment and the drugs, we can activate this very strong tumor suppressor called LHX6. We're also profiling some other genes as well. And our early results show us that there are some other very important tumor suppressors that are also being activated. It doesn't activate every single thing, but what we're looking into is whether the group of tumor suppressors that are being activated are sufficient to give us a very strong anti-cancer phenotype. And then we looked at cell viability first, right? So this is just an assay to look for metabolic activity in the cancer cells. And so we can see a significant reduction of viability in cells that express our activator, right? Probably as a consequence of these tumor suppressors coming on. And then we did a control experiment where we overexpressed the protein just to make sure that the cells weren't, just that the expression itself wasn't toxic. We can overexpress a neutralized protein, right? Without the activation domain, and these cells are perfectly happy. So we believe that there is a specific response coming from the synthetic activator. So we are already thinking about translation, right? The first one question that we get a lot is, how do you get the protein into the cells, right? Gene delivery, as I already showed you before, is going to be very hard to do. And we do that for our experiments in the lab for the foundational research. But to move this to translation, we really got to start thinking about delivery. This protein is very small, and I like to brag that it's smaller than CRISPR. And scientists already have taken CRISPR complexes and put those directly in the cells. Our protein is it's about 5 times smaller than CRISPR. So it's nice and compact. And we believe that if we take purified protein and add a signal like a cell penetrating peptide, we might be able to deliver this protein directly to tumors. So we've actually tested that idea out in 2D culture, and the uptake is beautiful, and it happens. You can actually start to see uptake in an hour. And then so what we did here was expose these cells to purify, right? So this is suspended in PBS, purified protein, no DNA. And then when we expose the cells for 24 hours and then wash away all of the non-incorporated protein, we can see really strong uptake in pretty much 100% of the cells. And we can actually control uptake by adding different dilutions, right, different doses to the growth media that's on top of the cells. We've also tried this in a cell culture that might look a little bit more like a tumor. And while the uptake is not as complete as what we saw with the monolayer of cells, these results are very promising. We are seeing a dose response. And so we're working on modifying the protein and testing some other time points to see if we can see improvement of uptake throughout this this tumor spheroid. Okay, so I'm going to, again, for the sake of time, skip a slide. We are interested in improving the strength of activation, right, of the fusion protein after it gets into the cell. So in order to do that, one of the function of the protein that we're focusing on is the binding domain. And we are interested in testing 3,000 different variants, right? So in order to test all of those, we have to have a really efficient system to put all those fusion proteins together. So we developed we customized our own Golden Gate assembly for fusion proteins that introduces just a 2 amino acid linker in between every module. So these modules can be anything. These are not just for our fusion. And excited to show in our hand IO and base repository are plasmids, right, to support assembly or add gene. Okay, so here's our vision, and it's not, these chromatin proteins aren't just misregulated in triple negative breast cancer. You'll see a similar situation in lots of different development related diseases. So our vision is to go and identify different actors in that pathway, isolate those, and build new regulators to try to treat the disease by re-regulating or sort of reprogramming the gene expression profile. So we are thinking about developing a whole catalog of parts to build chromatin proteins for epigenome actuation. And then for triple negative breast cancer, I've already started collaborating with some other scientists. We're working on using a more sophisticated model, cells grown in 3D. We're calling these, or they're called TMDC spheroids. We're going to actually take a look at what's going on at each individual cell using a very nice molecular imaging approach. And then what we're planning on doing is introducing in these actuators and then looking at the changes of phenotypes at the single cell level. Okay, with that, I'd like to thank our support, our funding, and all of the collaborators that have contributed to this project. Thank you very much.
Thank you very much, Dr. Karmella, for such an interesting